Hi everyone. In the previous lecture, we started with transcription in eukaryotes, where we have seen the types of RNA polymerases in eukaryotes, the promoter elements, initiation of transcription in eukaryotes with the role of general transcription factors. In this lecture, we'll begin with structure and production of eukaryotic mRNAs, where we will see the structure of eukaryotic mRNAs, and we'll compare the production of mRNA in bacteria and eukaryotes. This is the general structure of mRNA found in both bacterial and eukaryotic cells. Yes, there are some similarities in the mRNA of bacteria, that is prokaryotes, and eukaryotes. And there is a misconception many a times that the initiation codon is right in the beginning of the mRNA here. Let's say in bacteria, if I take example of bacteria, we know the initiation of the first codon is AUG, that codes for formal methionine or methionine. So the initiation codon starts right at the extreme. This is a misconception. No, the initiation codon may be somewhere here. So it's not at the extreme 5' end. Because in both bacteria and eukaryotes, there is a region in the mRNA at the 5' end called as untranslated region. This region does not get translated into the protein. This is the 5' untranslated region, or abbreviated as UTR, 5' UTR. UTR stands for nothing but untranslated region. This 5' untranslated region is also called as a leader sequence, and it's at the 5' end. Also, there is an untranslated region at the 3' end called as 3' untranslated region, or 3' UTR, or also called as Taylor sequence. And in between, there is a protein coding sequence between these UTRs, and this protein coding sequence specifies the amino acid sequence during translation. So these arrows represent translation start, where the ribosomes will begin the translation, and translation stop, where the translation will stop. So this is the protein coding sequence, and it is flanked by two UTRs, 5' untranslated region and 3' untranslated region. This is the structure of mRNA in bacteria and eukaryotes. Let's talk about production of mRNA in bacteria and eukaryotes. Yes, we have seen the process of transcription in detail in the previous lectures in both bacteria and eukaryotes, but let's compare the transcription process or the production of mRNA in bacteria and eukaryotes. So what are the differences in production? This is a prokaryotic or a bacterial cell. In this bacteria, there is a circular double-stranded DNA chromosome. For simplicity, I have drawn only the part of DNA. And here transcription is taking place by the RNA polymerase holoenzyme of the bacteria, which we have seen in the previous lectures. And this green color thing is the mRNA, which is being synthesized due to the transcription of this gene by RNA polymerase. In bacteria, the mRNA which is made is final. That means it is ready to be translated. Or in other words, I would say that the base pairs of a gene are collinear with the bases of this mRNA which will be translated. So it doesn't need any processing or modifications. Once it's transcribed, it's ready to be translated. It's final and it's collinear with the gene. Another thing is, we know our basics that prokaryotes doesn't have a true nucleus. This DNA of bacteria is present in the central region called nucleoid, but prokaryotes doesn't have the true nucleus, which means when this mRNA is being transcribed, it is in contact with the cytoplasmic region. And this purple color thingy are the ribosomes. So these ribosomes have begun the translation before the mRNA is completely transcribed or the transcription is completed. So since prokaryotes doesn't have a true nucleus, the translation begins before the transcription is completed or this mRNA is completely synthesized. And we can clearly see the ribosomes have begun the translation in this picture, even when the transcription is not completed. This is a hallmark feature in bacteria, and this is called as coupled transcription and translation. So where the transcription and translation can take place simultaneously. So we have seen the 2 points in the production of mRNA in this slide in bacteria. Let's see how the things differ in eukaryotes. This is a eukaryotic cell, animal, plant, or human cell. Eukaryotes have a true nucleus, which is evident in this picture, and the cytoplasm is separated from the nucleus. Eukaryotes have linear chromosomes. Again, only part of a gene is drawn here, or part of the DNA is drawn for simplicity. In eukaryotes, when the mRNA is made, it's not called as final mRNA first. It's premature messenger RNA or pre-mRNA, which means it is not final, it's not mature, it's not ready for translation yet. That's why it's called as premature or pre-messenger RNA or pre-mRNA. So this has to be processed to the mature mRNA. And that's why, in contrast, when what we said a few minutes back for bacteria, that in bacteria, the transcript which is made, the mRNA which is made, doesn't need any modifications or processing. It's ready for translation and it's final. In contrast, eukaryotes, first pre-messenger RNA is made. So this pre-mRNA will be processed. You can see a circle here. This is addition of a cap, and poly A tail is added. And further it is processed. We'll see this processing in detail in the next lectures. And this pre-mRNA is processed and converted to a mature mRNA, which is green in color or shown green in color in this picture. This is called as RNA processing. The process of conversion of premature messenger RNA to mature mRNA is called as RNA processing in eukaryotes, and it happens in the nucleus. In eukaryotes, the mRNA has to be transcribed completely and processed before it can be translated. You will not see coupled transcription and translation in eukaryotes, and it's not possible. The reason being is that ribosomes which are involved in translation are located in the cytoplasm. So the mRNA has to be, transcription has to be completed. The mRNAs have to be processed, the RNA processing has to be completed, and then the mature mRNA has to be exported from the nucleus to the cytoplasm for translation. So these are the two major differences in the production of mRNA in bacteria and eukaryotes. Let's see a third one. Another fundamental difference between bacterial and eukaryotic mRNAs is that bacterial mRNAs often are polycistronic, meaning that they contain the amino acid coding information from more than one gene. What do we mean by polycistronic mRNAs? In bacteria, the mRNAs often are polycistronic. Let's say this is a bacterial mRNA and it's a polycistronic mRNA. It has amino acid coding information from more than one gene. So let's say 3 or 4 genes have been transcribed together, giving rise to this transcript or mRNA, which is polycistronic. And this mRNA has the amino acid coding information for those 4 genes, which means it can code for 4 polypeptides or proteins. Why this happens in bacteria? We know what are operons. Operons are cluster of genes with a single promoter and a common operator sequence, and these genes are regulated together. They are transcribed and all the proteins are made, or none of them are transcribed and no proteins are made. So this is an example of a polycistronic mRNA having the amino acid coding information for more than one gene and coding for more than one protein. And bacterial mRNAs are often polycistronic. In contrast, eukaryotic mRNAs typically are monocistronic. I'll use a different color here. So eukaryotic mRNAs, as said, in contrast, are monocistronic. That is, they contain amino acid coding information from just one gene and code for a single polypeptide or protein. So in this lecture, we have seen the structure of mRNA and how there are similarities in the structure of mRNA of bacteria and eukaryotes, untranslated region at both ends. And then we have seen the differences in the production of mRNA in bacteria and eukaryotes with some of the major differences.